Hello, my name is Akir Pimenov. I do my research with Dr. Hank Postma here at Postma Labs, and today I would like to present my research on towards single molecule DNA sequencing with graphene nanogaps. In recent years, the advancement of DNA sequencing techniques has offered a new frontier in biotechnology and has furthered our understanding of genetic diseases, ushering a new era of personalized medicine. Despite the completion of the Human Genome Project, with its ambitious $1,000 cost target for the whole genome sequence, there is still no technique available that can accurately sequence the entire human genome at this cost. And this is where my research comes in. Here, we have developed the technology to create graphene nanogaps capable of reading DNA's transverse conductance. Our approach allows us to implement a high-frequency electronic interface in conjunction with a graphene nanogap that allows us to simultaneously and rapidly read long, single-stranded DNA with minimized noise through its transverse conductance. Before we can talk more, I would like to explain some basic terms. First and foremost, DNA. DNA is a nucleic acid containing the genetic instructions used in the functioning for all known living organisms. DNA is stabilized by two backbone helixes that are negatively charged, and they're held together by the base pairs. Each base comes in a variety of form, being C, G, A, and T. And for the purposes of our research, we cannot operate with double-stranded DNA or like natural state of DNA. We have to denature the DNA and turn it into single-stranded. This can be done with various methods. Three of the most common ones is chemical melting, where we introduce an additional chemical, thermal melting, where we change the temperature, or the lowering of the salt KCl concentration of the DNA mixture. By doing this, we're able to stabilize the single-stranded DNA and use it for a DNA translocation. Important things to note that the diameter of the single-stranded DNA is approximately 1.1–1.3 nanometers, while the distance between each individual pair of bases is about 0.33 nanometers. This nanometer size scale is primarily the reason why for transverse conductance you need such thin electrodes, and this is where graphene comes into play. Graphene, otherwise known as a single atomic thick layer of graphite, as you can see here, is an amazing material for our electrodes. Not only is it intrinsically conductive, it also can withhold high transmembrane pressures. In the picture, you can see various thicknesses of graphene by layer. The darker areas have more graphene layers on them. The lighter areas attribute to a single sheet of graphene. You can identify this optically, and this is how we're able to tell how many layers of graphene we use for our research. Now that we got those two basic properties out of the way, let's talk about why nanogaps over anything else. Graphene nanogaps allow for a direct read of long single-stranded DNA. Using the graphene as an electrode, we're able to actually read each individual base. That is, we are able to do a translocasing sequence even if the bases are all the same, for example, TTTT or AAA. This is known as a homopolymeric sequence, and with the use of graphene nanogaps, we will be able to achieve single base resolution. With the conjunction of nanogaps with our wafers, we're able to produce very cost-effective and very small devices on a wafer scale. This makes it very portable and commercialized. Now let's talk about the devices themselves. On the picture right behind you, you see the silicon nitride wafer. On the silicon nitride wafer, there is a membrane. In the membrane itself, there is a micropore. The micropore is situated right in the middle and is going to be the tunnel for which the graphene is going to transverse, and this is where we ideally would like to put our graphene, and we will make our break in the graphene, aka the nanogap, over. As you can see here, EBL, or electron beam lithography, was used to etch a resist onto our wafer, where we will deposit gold to make a gold electrodes leading to the graphene, and we will position a graphene flake right over those electrodes. Right here, you can see the actual construction of the gold. The gold is deposited. Now the gold electrons have been set, and the graphene has been positioned right with the gold electrodes. Now, to make sure that there's no leakage, that the graphene perfectly seals the membrane and the micropore is completely covered and our electrodes are properly connected, we have to actually now assemble our wafer. This wafer assembly is done entirely underwater in our microfluidic cell. The microfluidic cell gets the wafer placed directly in the middle, and it gets submerged into water. This is done to avoid any bubbles, even micro and nano bubbles. These bubbles, if found, will cause us to misread the resistance of the ionic current, which will lead us to have faulty results. And so it's extremely crucial for us to submerge our devices and make sure that we have no interference. After everything's connected, we are able to measure the IV characteristics, and we should see a completely isolated system. So there's no current going through because the graphene has yet to be broken. To actually facilitate the breaking of graphene to make the nanogap, there's two primary ways that we use in our lab. One is via capillary-induced breaking. That's where we use the capillary forces, knowing the height of the gold and the distance between the gold electrodes. We're able to use capillary forces to break the graphene and cause a very specific gap in terms of its distance. This method has been used in the lab in the past. However, now we've transitioned more to dielectric breakdown, where instead we use the varying volts pulses to induce the break in the graphene sheet. This can be done by our computer, and we set specific voltage ranges to make sure that we don't go overboard and we construct the ideal graphene nanogap. After the nanogap is created, we run our IV characteristics, we measure its resistance. Via that, we can double check to see the size that we got and if everything goes according to plan. For a few beginning setups, we can also see optically to test if we have the gap. If everything is secure, we can now flush our microfluidic cell with the DNA and proceed with the translocation. As the DNA is getting flushed into the cell, we apply a positive voltage bias. Since the backbone of the DNA is negatively charged, it's going to drive the DNA through the micropore and through the graphene nanogap. As the DNA is getting driven through the nanogap, we measure its ionic current, or more precisely, the blockage of the ionic current. Every time the DNA goes through, we will see a drop in the ionic current. That's why we are able to identify that DNA is going through and we're actually reading translocation. That, however, is not the prime use of the nanogap. The main point of the nanogap is the transverse current. The transverse current is the current that goes across the graphene through the DNA to the other piece of graphene through the quantum principle of electron tunneling. This will also be notated as a voltage difference, and due to the single base resolution, we should be able to identify each individual base pair. However, to get to that, we must have our setup perfect in terms of our signal to noise ratio. And this is where the next step of my project comes into play. Right now, I am in the process of making and finalizing a current amplifier that will amplify the signal so we are able to read specific signals that we receive with a high enough accuracy that the signal doesn't get overshadowed by the noise, and we can tell the difference of each individual basis. This involves making sure that our noise levels are as low as possible, we have an appropriate bandwidth, and the signal to noise ratio is maximalized. Lastly, I would like to thank my coworkers, Diana, Robin, and Wolf, my current lab tech, Ravipa, and my fellow undergrad, Arceli. Additionally, I would like to thank Professor Hendrik Postma and the National Human Genome Research Institute, because without them, this would not be possible. Thank you so much.